This is the third session of the Viromics Workshop, the third and final session on virus databases. I think it's been a fantastic workshop so far, so really thanks to all of the speakers, and I'm excited about this session as well. As a reminder, this session has a slightly different format. It's still 1 to 3:30 p.m. Eastern Time, but we have a shorter introductory talk and some slightly longer database talks here. All right. So an introduction to virus databases, that's what I'm here to do. I wouldn't say that virus databases specifically are my area of expertise, but it's something that I'm very interested in as a user. So my lab studies soil viral communities. And so sort of the slant that you're going to get in this introductory talk is much more the slant of a group that is interested in using virus databases. And then, of course, the rest of the talks are the experts who have been involved in developing some of these fantastic databases. So I'm going to go over some potential use cases. Why do we need databases? Then some brief examples from a study from our group on peat viruses, where we used a database to do some analyses. And then at the end, I'll touch on some considerations and caveats for choosing a database and then evaluating the associated database output. Full disclosure, those are some really wordy slides. Those would be good for, a screenshot if you don't catch all of it. Screenshot any of this is totally fine by me. So what do we mean about a virus database? I think there are lots of different kinds of databases that could be involved in viral analyses. But in the context of this talk, I'm mostly talking about a set of viral genome or maybe context sequences that are going to be associated with some metadata, very broadly defined. So a little bit of housekeeping. During Manja Marz's talk, she's going to introduce you all to a really cool document that she's putting together on what users want from a database. So at that time, you'll have the opportunity to click on that link that I'll put in the chat and add your thoughts. So be thinking about what you want from a database as you're watching some of these talks. So one use case, all right, that's what we're going to do for the next few slides. This is a use case from metagenomic data, let's say, definitely slanted towards soil viruses, but this could be any data set. So we're going to collect our own samples. We're going to extract and sequence, shotgun sequence some metagenomic or viromic DNA. We're going to do some de novo assemblies, maybe some viral predictions on our own data so that we can get our de novo viral sequences. But then we also want to incorporate sequences from a reference database. So we're going to add those reference viral genomic sequences. This can be, a database of your choosing. Then maybe we want to cluster all of those viral context together at the VOTU level. Maybe that's at 95% average nucleotide identity, so that we now have a reference set of sequences that are both de novo assembled and from public databases. Now we can use that reference set to map our reads back and get a feel for which viruses are present in our sample, including those that didn't de novo assemble. Okay, so here is our read mapping, again, to get the presence of those viruses, maybe from the public database. So that's one use case. Another use case is performing a meta-analysis. So you can do this meta-analysis on the database exclusively, or maybe you can add your own sequences to the database to do a meta-analysis. But the example here is from the Paez-Espino et al. Earth's Virome study that was published in 2016. And so the idea here is that they're comparing the viruses that were recovered from these different environments, which are shared, and how do they compare. So you can leverage databases to run some kind of meta-analyses. You might also want to place your unknown viruses in some kind of context. In this case, this is a phylogenetic context. So this is an example from a soil metatranscriptomic data set where the authors, Evan Starr et al., went and mined metatranscriptomes for RNA-dependent RNA polymerase sequences, and then made a number of phylogenies that included reference sequences from either databases or published papers, and then also included their own sequences so that they could get a feel for the phylogenetic spread of the RdRps that they recovered relative to what was known already. So placing your unknown viruses in some kind of context is also a good use for databases. Assigning metadata to new viruses is one possible use case. So what do I mean by that? This is an example of actually using an earlier iteration of vContact to assign taxonomy to new viruses based on their clustering with old or previously known viruses. And so there are lots of caveats associated with how close you need to cluster in order to confidently leverage that metadata and make those assignments. Not going to make the case here of exactly what those thresholds and cutoffs should be. But the point is that you can leverage the metadata that exists in these reference databases to try and add some additional information about maybe some unknown sequences in your own data. So this is just a network that we ran from a thawing permafrost data set in 2018. And we have our quote unquote unknown sequences. In yellow here are unknown viral context sequences. In this case, they were essentially VOTUs. And then we have RefSeq sequences and a number of other publicly available sequences. And so the closer our sequences cluster to those previously known sequences, that might help us to think about assigning some metadata that was already publicly available to our unknown sequences. So you could also use databases to benchmark or train a software tool. If you're developing a software tool, pretty much all software tools are going to have to be benchmarked or trained on some kind of data set. So if you're not developing, developing software yourself, you might interpret results from a software tool that was trained on a specific database, or from a software tool that's going to use a specific database when generating output from your data. So this kind of ginormous wall of text here is meant to explain some of the things that you might want to think about. So you might want to consult papers or GitHub for a given software tool to understand which databases were used when that tool was developed and how those databases were used, to determine whether any databases are going to be used with the software alongside your data to try and identify signals in your data. And then the point of doing all of that is to try and use that information to assist with the interpretation of your results, so that when you get that output, you can think about whether there are any strengths or limitations based on the chosen databases that were either used to develop the software and or used alongside your data to make some predictions. So now moving to some real data. Those are just a few use cases. There are many more possible use cases in this wonderful document that Manja is going to share later. But this is a set of analyses that we ran on some data from thawing, or not thawing permafrost peatlands. So used to saying thawing permafrost peat. These are actually very different peat. These are boreal peatlands in northern Minnesota. They're collected right here. And the idea was to leverage both this de novo metagenomic data set that I'll introduce you to, and a database that somebody named Anneliek ter Horst in our group had developed. That's a combination of these de novo sequences and some reference data that I'll show you in the next slide. But first, to introduce you to the data that we collected. So this is a bog in northern Minnesota. This is a beautiful transect through that bog. We collected some cores along the transect, looks like that. So here is the bird's eye view of where we collected our samples. From our own data, we only had 5 samples, so we had 5 viromes and matched metagenomes from the same samples. But it turns out that the DOE has this awesome experiment associated with this site, and the JGI, in collaboration with some of our collaborators, had already sequenced 82 metagenomes from these experimental units where they were looking at manipulating carbon dioxide and temperature. So they had 82 metagenomes in combination with our 5 viromes and metagenomes. And we ran some bioinformatic analyses, including VirSorter, to predict VOTUs, essentially to predict viruses from these genomes or metagenomes and viromes. Got about 4,000 VOTUs. But these VOTUs were recovered from a combination of de novo assembly and mining of these data and from a database that Anneliek in our lab had compiled. So I'll introduce you to that database now. This is actually not a plug for using our database. It is publicly available. You're more than welcome to use it if you like, but it's much more, we wanted to compile a database that met our needs. So you can go to these publicly available databases and then add your own data, add data from other papers that are of interest. So we kind of wanted to bolster the soil part of our database. All right, so we compiled these VOTUs essentially from a number of different ecosystems in this database called PIGEON, or Phages and Integrated Genomes, Encapsulated or Not. And the idea was to do 2 things, 2 of these use cases that I've described before. So one is to pull out more sequences from our unassembled data. And 2 was to do a meta-analysis to kind of place some of our soil viruses in context. So first actually is the meta-analysis. So this is now based on vContact2 viral clustering results. So we're looking at quote unquote the genus level here. We're looking at what viruses were shared across these different ecosystems at the viral cluster level. So you can see that from the PIGEON database, most VOTUs were exclusive to the different environments. So if they were found in soil, they tended not to be found in marine and freshwater environments, and vice versa, although there were some VOTUs that were shared across the data set. And I should say these are VCs that were shared, and they were shared because the VOTUs that went with those VCs were found in more than one environment. All right, so you can do a meta-analysis. And then we also wanted to kind of get a feel for some context for our data. So which of these soil viruses had been seen previously and which were found only in northern Minnesota? So this is based on read mapping to the PIGEON database from our metagenomic reads. And what we're looking at here is the locations from which each of the VOTUs that were recovered in northern Minnesota were initially found. So here, to orient you, these are the VOTUs from northern Minnesota exclusively. So 4,126 were de novo assembled from our data set and not seen anywhere else previously in the PIGEON database. But on the other hand, these 147 VOTUs, which is why I keep saying thawing permafrost peat, these are actually from some of our earlier studies in Matt Sullivan's group, particularly with Gary Trubel. So 147 of these VOTUs that were recovered in Minnesota peat were actually previously recovered in peatlands in northern Sweden. So this is kind of giving us a sense of, how much are we starting to see the same kinds of viruses over and over again. 4% of the data set might not sound like very much, but we're kind of getting there. Maybe there's some hope for capturing the global soil virome if we start to continue to collect samples in a number of different locations. But also interestingly, as a result of this meta-analysis, you'll notice that there are no VOTUs in the oceans here that were recovered in Minnesota peat. So maybe there is potentially an aquatic or terrestrial species boundary. Remains to be seen. All right, so as I warned you, the wall of text with some considerations and caveats. If you wanted to take a screenshot, that would totally be appropriate. So I'll try and go through this quickly. I'm not going to read you a giant wall of text, but some things to think about when you're considering what database to choose or whether to choose one at all. Obvious question is, how useful is a database even going to be for answering your scientific question? So for example, from soil, almost everything that we recover is from our de novo assemblies, just because databases aren't super well equipped with soil viruses at this point. So maybe we don't even need a database, depending on your question. Are you prepared to read the papers and documentation to get a feel for why or how the database was developed and why it might or might not be useful for you? What data sources do you want? Like, do you want RefSeq, isolate genomes, genomes from metagenomes, etc.? What metadata are going to be available for each of those instances in your database? So for each viral genome, for example, what kind of curation has been done on the database already? And is that going to work for you? How much do you care about redundancy in the database? Do you need your VOTUs, for example, to be clustered or not? And then, of course, tractability. Is the database too big for the computational resources that you have? And if so, does the database itself somehow offer some options for culling the data to a size that will work better for you? So in addition to caveats for choosing a database, there are, of course, caveats for the analyses that are going to be associated with a database. And I'd say the biggest one here is to watch out for false positives. If you have a database that has lots and lots and lots of viruses, including high and low quality, and you start to see some low quality viruses in your data, it's kind of this garbage in, garbage out idea. That doesn't mean that databases that have a lot of sequences are bad. It just means that you need to very carefully consider what parts of the database you're going to use and trust. So watch out for false positives. Of course, true positives might also be missed. A classic example from our data is that we use a stringent 10 kb cutoff in genome length. And of course, there are viruses that are shorter than 10 kb. You want to keep track of different database or software versions because your output can look different depending on what version you're using. And consider manual curation of your data. All right, so I am going to mostly stop there. I do want to highlight that NCBI is not a part of this database discussion, and that's because we want to introduce you to some new stuff that hasn't been in some of these kinds of discussions recently. But if you do want to check out NCBI, you can check out Rodney Brister's talk that was recorded in 2018. I just Googled Rodney Brister NCBI JGI, and that got me straight to his talk. I'd also be happy to put this link in the chat. And in the interest of time, I won't take questions at this point. I'll happily answer them in writing, but I was kind of planning to go straight to the next talk anyway.